All right, well, welcome everybody to our virtual sensory session on yeast nutrition. I'll have just a couple of announcements to start off today, but we do have a full schedule of great things to share with you. We have Dr. Amanda Stewart with us today. She's going to start things off by giving us a brief overview of some things to know about yeast nutrition, and then we're going to open the floor to your questions. We've put in a good chunk of time for questions and answers about yeast nutrition. Then we will move into looking at the two different experiments that hopefully you received your samples for, that kind of hone in on a couple of these finer points of yeast nutrition. Before we get started, just a couple of announcements. You should have received the invitation for the April 1st sensory session, which is on sparkling wine base. So we'll talk about some ways of managing some strange acidity numbers that we saw this year in our sparkling wine base. And also the April 15 session looking at measuring and managing SO2. We'll have an experiment that's sort of the third part of some SO2 management experiments that have really changed the way I look at SO2 and I think have been really valuable for a number of people. But we'll also be talking about some experiments looking at different measurement techniques for SO2. So if you did not receive the invitation, it was one email with invitations to both. If you did not receive them, do check your promotions folder and your spam folder just to make sure we're not getting filtered out. But if you didn't receive them, please send me an email and we can figure out why my emails are not getting to you in case that's a problem. We are on the Zoom platform today, and as we've already talked about, that means that you control your own microphone. So we do encourage you to unmute yourself and participate in the discussion if you have questions or comments or something to add to the discussion. But we also thank you for keeping yourself muted when you're not actively participating in discussion, just so that we can cut down on the ambient noise. Thanks for those of you that are able to keep your video on. That makes us feel like we're talking to somebody other than our computers. But we do understand if you can't keep your video on, we do know it eats a lot of bandwidth, and sometimes there's some other issues with that as well. So that one is your choice. Do know that you are being recorded today. We will be recording our session and posting that on our YouTube channel. And if you have missed any sessions in the past and you want to go back and listen to any of those are all posted at this point on that YouTube channel. And then lastly, the chat is open today and you can feel free to add items into the chat at any time. I will try to be faithful to keep monitoring that as people are talking and be able to convey those ideas to our speakers or any questions that you have. So let's go ahead and actually use that chat to introduce ourselves. So if you can write into the chat what your name is, in whatever vineyard or winery or entity you're representing today. And just so that we can know who's going to be presenting today, let's maybe have our panelists introduce themselves. So if each one of you could just say who you are and where you're from, maybe Mike, you can go first.
Hi, I'm Michael Heny. I'm the winemaker at Michael Shop's Wineworks.
Great. And Rick?
I'm Rick Tagg. I'm a winemaker at Delaplane Cellars.
Okay. And Amanda?
Hi, I'm Amanda Stewart. I'm an associate professor in food science and technology at Virginia Tech.
Great. Great. And we welcome to each one of you. I see your comments starting to come in. We really are thrilled to see each one of you, and we're thankful that you're willing to take your time today to be with us. As you continue to kind of add those in, I'm going to go ahead and give Amanda a little bit more of a formal introduction and hand things over to her so that we can get started with our program. So Amanda Stewart, we just welcome. We're so thankful that you're here with us today. Amanda is a tenured associate professor of food and beverage fermentation at Virginia Tech in the food science department. Her work focuses on the impacts of yeast assimilable nitrogen composition and concentration, both on yeast and bacterial metabolism before during and after fermentation. Some of her more recent work looks at developing vineyard and orchard practices to improve wine and cider quality. She has some really interesting ideas about the roles of nutrients in terroir, so hopefully she'll touch on that a little bit as well. So Amanda, welcome. We're so glad you're here, and I'll hand the mic over to you at this point.
All right, thank you guys. I really appreciate the opportunity to talk to this group and to be part of this group today. I'm gonna share my screen here, and I've got a PowerPoint file on my slides are on my external monitor. So if you see me keep looking to the side, that's what's going on here. So yeah, so let's see. Do you guys see my slides? Can I have a thumbs up? You can see my slideshow. Cool. Thank you. Thank you. All right. So I am really, really glad to be here and talking to this group. As Joy already gave me a nice introduction, so I work mostly with fermented food and beverage, mostly cider, wine, and cocoa at this stage. And so I'm really, really excited to be getting into more wine work. And I'll mention a project that I'm working on now at the very end, dealing with wine. But I think most of you are probably familiar with Virginia Tech. We're a public land grant university, and I'm part of the Department of Food Science and Technology. We've got a growing emphasis on fermented food and beverage science, and we do research, teaching, as well as extension work in this area. And I'm sure you guys are all familiar with my colleague, Dr. Beth Chang, who's been doing enology extension full-time with us for a few years. So we as a group are very interested in partnerships in research, teaching, and education. Our fermented food and beverage program focuses on several areas. I'll bring up a few. We've got brewing and malting going on, got a great study abroad with the Technical University of Munich now. So it's like a 4-week study abroad. So exciting. This is a cocoa, picture cocoa germplasm repository in Puerto Rico. We're working with these folks on some cocoa fermentation and cocoa chemistry, cider research going strong in collaboration with other groups, and then also wine research going strong. This is some Cabernet Sauvignon from up at the Winchester AREC that Tony Wolf grew, and we did some research on. So my major research,
Amanda?
yes.
Amanda, I hate to interrupt. I'm not seeing your slides advance at this point. I just see your slide window. So,
wait.
Yeah, we're seeing your slide window, but not your slides advancing. I'm
no,
sorry.
Thank you for cutting in. I'm on like slide number 8 now, so I'm glad you mentioned that. So you still see my title slide?
We see your title slide, but we see your whole slide window. We don't see the presenter mode.
Here, let me fix that. Is that better?
Yes, we see your first slide and you've just advanced, so all set.
All right, so there's some nice pictures of the brewing technology, the cocoa plantation, not in Virginia, the cider work. We've got, this is in Virginia, this is at Winchester. And then that's the nice Cabernet Sauvignon that Tony Wolf grew up at Winchester. So, and we're doing some research on that there with some polyphenols work a few years back. So my major research themes in my lab have been on nitrogen and fermentation. Most of this work has been done in cider systems, and I'm really excited to be getting started on some more wine-based work in this area, mainly looking at how can we manage nitrogen to meet stylistic goals, and where does that strategy play in the overall framework of winemaking. We also have done a lot of work on polyphenols in both cider apples and wine grapes from a product quality and sensory standpoint, as well as value-added products from byproducts and foods and health. So those are my main areas of research, and I'm going to spend the rest of the time today, hopefully a little under 20 minutes, talking to you about the nitrogen theme and just some thoughts. And I think I'll probably bring up more questions than answers, just to be upfront about all this. So, you know, we want to talk about yeast nutrition requirements today. So the major requirements for yeast, of course, are sugar, nitrogen, then other micronutrients like vitamins, minerals, and lipids, and also a lack of biotic and abiotic stress or threats to the yeast. So all these factors play into how well yeast do during fermentation. It's not just, you know, a DAB number. And I want to emphasize that throughout. So we want to talk about YAN, just to all start out on the same page. What is YAN? It's yeast assimilable nitrogen, it's an acronym for that. And it's the nitrogen that's available for yeast to use during fermentation. It includes ammonium ions. So that's the A in DAP, diammonium phosphate. So ammonium ions, free amino acids except proline. Those are the major components. And then also probably some small peptides, though those are a little harder to measure. It does not, we mostly agree that it does not include proteins or very large peptides because yeast cannot bring those into the cell. So why does YAN matter? Well, first and foremost, the lack of YAN can lead to stuck or sluggish fermentation. And this chart is from a paper from a group in Portugal, but it just shows that, if you have, this is the bricks here on the vertical axis. And can you see my cursor? Okay, yeah, so this is the bricks on the vertical axis, or the sugar is glucose in that case. And this is hours. And all this is showing is that different YAN levels, you're going to get different fermentation curves. And when there's very little YAN, like less than 60, this is milligrams per liter, but it's similar to parts per million. Many would say the same. So these values are not going to be enough to have a fermentation take place. Then, they showed with 260, that was enough to bring the bricks down to dryness eventually. And then with more and more, it sped up. So, definitely YAN affects fermentation rate. This is a very old chart from 1979. It's long been known that there's a relationship between hydrogen sulfide production in wine fermentation and YAN concentration. But you can see here, this is the hydrogen sulfide production on the vertical axis and the YAN value on the horizontal axis. You can see there is a general trend, more YAN, less H2S, but there are, it's not a perfect correlation. There are clearly other factors that are involved in this relationship. And I think that you guys from Making Wine will all appreciate that. But in general, that is one of the reasons why we want to add YAN. So before we talk about addition strategies, I wanted to take a minute. I had a lot of fun preparing this presentation. I got to look back through some slides that I've used over the past 10, 12 years. I used to do a lot of winemaking extension when I was in the Midwest, when I was at Purdue. So I gathered some data during my time there, and I hadn't been able to use it a lot since. So it's a lot of fun to be able to kind of share this story. So I want to make the point about how much YAN is naturally present in wine grapes. So this is a survey that I did when I was a grad student, and it's got, we did over like 1,400 samples, and I gathered data from some other colleagues, like people at Cornell, at Purdue, at University of Kentucky. I mean, just all over. I did kind of a YAN survey of these lesser known wine making regions to see where we were in YAN. And these are the grape varieties on the horizontal axis. And you can see we've got some muscadines, all kinds of hybrids, vinifera. We just got it all, just like the Midwest has with winemaking. So I took the 64 different varieties, and I just grouped them by variety on this plot. And you can see the YAN values. That's what's here on the vertical axis. They vary a lot. I think I've got some. Yeah. So the muscadines that were grown in Florida, I got some Florida samples in here. They were way at the low end in general. And the hybrids developed by the University of Minnesota were way up here on the high end. And they ranged all the way from like 100 up to like over 400. And that's the average for that cultivar. So we had some single samples way up in the 700s, even from like Iowa. And you think about it, super rich soils, places that used to be growing soybeans, there's going to be a lot of nitrogen in the soil. So like tremendous variation. It's a starting point just to say, where are we at? So I took that same data set and grouped them by kind of broader categories of like hybrids. This was Norton, Labrusca grapes, muscadine here, and then vinifera. And then I put an apple here. Ken Hurley and Ann Sandbrook gave me some apple data when I came to Virginia Tech, and we grouped that in here too. And just in general, you can see there's some hybrids are higher in general, and the Labrusca, and then the muscadine's lower. The vinifera is coming in, somewhere around 200, which makes sense. And the apple was way lower than the rest of these. Some of that could be just an artifact of the places where these are grown, like the Florida plantings being in sandy soils with really a lot of drainage, not a lot of organic matter, the hybrids being grown more in the Midwest and new plantings and that. So I'm not saying that this is necessarily an effective cultivar, but just mainly wanted to demonstrate how much it can vary and just help people get their head around whether they're really dealing with a deficiency in general or not. So there is also some data from some previous published yen surveys here. These are from the West Coast of the US, and they saw ranges from 40 to 550. And then another survey saw from 25 up to 336 milligrams per liter. And then my survey was showing about 100 to 400. So the point is that yen concentrations are highly variable. So they can vary a lot. And the composition there, this is a plot that shows the amino nitrogen or the ammonium nitrogen plotted against the amino nitrogen. And if there was any relationship between those two, like let's say if the ammonium was always double the amino or vice versa, that line would be straight and there'd be a really good correlation, but it's not. It's a scatter shotgun. So some samples might be super high in ammonium and low in organic. Others might be the opposite. So all this data basically has led me to recommend that people measure the yen and understand what's there, at least until, maybe if you have your vineyard management, your vineyard very well established, management practices very well established, it might not change a lot within a given cultivar from year to year. Rainfall can affect this too. So, it might still change some, but it might get dialed in. But especially if you're working with a new vineyard or with new growers, if you're winery buying fruit, it might be good to measure this because it could potentially vary a lot. So I think I want to make the point right now that I think single target yen numbers are problematic, and they're hard to, I think the industry has maybe pushed the suppliers from the enological products industry has at times pushed targets or maybe equations to calculate targets. And I think it's hard because there are definitely factors that interfere with the amount of yen we need. We know different yeast strains need different yen values. That's good. I think we can recognize that and all agree on it. But I think also the relative concentrations of the amino nitrogen and the ammonium nitrogen are gonna have an effect on the fermentation outcomes. And the amino acid composition can also affect that. And I think it's pretty well established that other factors can interact with it, things like vitamin concentrations like biotin and pantothenic acid, some other micronutrients. If they're not there, you might be able to add a whole bunch of yen and overcome that lack. But if they are there, maybe you don't need as much yen to get through the fermentation without H2S. So I think that it's a really complex relationship, and it's frustrating. But I think it's more complicated than just a target value. So I want to talk about a few of those factors with the composition now. Moving forward. So amino acid profile in juice, certainly it's been shown in the research that wine aroma can be influenced via the effects of amino acid composition on yeast metabolism and the esters they might make during fermentation or might not make. So juice nitrogen composition does impact wine aroma. It can, certainly, to a greater degree in some cases than others. And also, I think maybe some amino acids might directly affect the wine flavor. It's been shown a lot in a tomato sensory evaluation, for example, that savory and bitter flavors can emerge from amino acids, and that glutamine can be a flavor enhancer, just like MSG is in cooking. Glutamine at the levels that we see in tomato, which aren't so different than the levels we have seen in some wine grapes, some of the hybrids especially, it might be making a more of a savory flavor or different kind of a flavor enhancer role. And that hasn't been studied much, but I think it probably should be, and it might be worthy of more investigation. So just to summarize, there's a few papers out there on studies just like what we're going to taste today with the different yeast nutrients and wine aroma. And in general, I think those few studies lean toward with more DAP additions, you may get decreased sulfur-like aromas and maybe a little more citrus character. With amino acid additions, you might get decreased sulfur-like aromas and then maybe richer in fruity notes. So, the question is, does the yen composition affect the varietal character? I wonder sometimes is if you add a bunch of nutrients, are you going to change or alter the varietal character? And if that's what you want to do for stylistic goals, fine. But if it's not what you want, then maybe DAP is fine, right? So the question remains: can nutrient additions help us to meet stylistic goals? And I think, I would say maybe, likely, in certain cases. But what's the potential downside? And I'd like to share these data as I wrap up here, because one of your other studies is on like the goal of creating a nutrient desert and trying to target just what you need and not more. And so I think that's probably because we're worried about the consequences of excess yan and like encouraging things like, film yeasts or just unwanted bacterial growth or Brett growth that we don't want. So, it's tough because we want to add these nutrient products that add nitrogen and maybe other micronutrients, but how do we recognize if we have a micronutrient deficiency? We don't measure those things in winemaking all the time. So it's tough to kind of match the right nutrient to the right winemaking situation or the right juice or must when you don't know what the deficiencies are and we don't have a real good handle on the benefits. We want to also consider the potential drawbacks. So, you could, like I said, produce unwanted microbial growth. And I think it could also add some savory or umami notes in certain cases with really amino acid rich nutrients. It might change things that happen in chemistry during long aging periods. And it might alter the varietal character. So one thing that I did this when I was a grad student back at Purdue, and I know, Kirsty, I see you on here. I remember you came and judged at this wine competition many years ago. And I was thinking of this data set, which I haven't used much lately, but we had this question. And so I was a grad student at the time, and I took a bunch of the bottles that had been opened for this competition and analyzed the yan in the commercial wines. So I did 128 wines, measured the ammonium and the NOPA. And so these are the values that I got, and I grouped them by grape variety. I mean, some of these, like the Albarino, I just had one, so there's no error bar. Some of the others I had, there are probably a lot more Cabernet Sauvignons, but there are at least two of any that you see an error bar on. But anyway, the point is it varied a lot. We had some with, hundreds of milligrams per liter of residual nitrogen left. Let me go back once, of residual yan left. So, some of them were really low. There's, you would call this probably close to a nutrient desert, some the Tannat, this Malbec down here, but then some of these this Marquette was super high. That's a Minnesota hybrid. Maybe it started out really high and somebody added nutrients and then it just, it's way up there. So probably they didn't need to do that. The same data set here is grouped by those broader categories again, and then you can see the hybrids had a lot more left over. And one of the things we struggled with a lot there was microbial stability in wines and getting people to bottle promptly and keep the sulfites up and top up and just basic things. But I think that this may have exacerbated their challenges with wine stability, like adding nutrients when you're already really high yan. So that was kind of the perspective I came from in collecting this data, but I thought it'd be fun to share it since we're talking about residual yan in this session. So the composition to it, this just shows what that residual yan was made up of. And basically it's the same thing as before in the juice. It's not clear. Like sometimes it's mostly amino nitrogen, sometimes it's mostly ammonium, sometimes some of each. So there's like no correlation. That's what this value means. That basically if that value was 1, it would mean that they're totally correlated. 0 means like it could be either one. So I think that this points to the fact that managing nitrogen in winemaking is sort of an art and a science. Definitely, I believe that the origins of wine style are influenced by nitrogen. The fruit has amino acids in it that are aroma precursors. Yeast metabolism is going to give us, esters, which are generally good. Acetaldehyde, maybe not so good. H2S, we can probably mostly agree is bad. And then the microbial growth and other yeast and bacteria growing in there, giving us some off flavors that we might not want. And, too, we could see some effects on oxidation as well by just, promoting microbial growth and using up oxygen. So I just want to put, to kind of wrap this up, where does this fit in? I really, really think that nitrogen plays a role in terroir, maybe a kind of an unseen role in this complex relationship between the vineyard management practices, the cultivar, and the wine style. But I think, we need to keep it in context. I don't think it's the key factor. I think, when you decide to make wine, you're first going to decide what's the wine style, what grape variety, what harvest maturity, what condition is the fruit in. All those things are playing a bigger role. And then your processing plan, your barrel aging plans, yeast strain selection, fermentation temperature, and then after all that, the nitrogen management comes into it. And I think it's worth thinking about, and it's worth, I hope it's worth doing more research on, but I think, all these other factors are playing perhaps an overriding role in wine style determination. So I'm going to just end this by mentioning this new project. Tony Wolf and I are both PIs on this larger project that is funded by the U.S. Department of Agriculture Specialty Crop Research Initiative. And right now we've got a group of, I think it's like 6 universities. It's led by Washington State, Virginia Tech's on there, UC Davis is on there, Cornell, Oregon State, Rochester Institute of Technology, and it's a precision ag project, which is looking at high resolution vineyard nutrient management. So looking at how we can manage vine nutrition and maybe correlate spatial imaging. It's really more driven by ag engineering, but it's looking at managing vineyards at kind of a smaller resolution using the spatial imaging. And so Tony and I are working on correlating fruit chemistry to tissue analysis and then tying that back into what the ag engineers at Davis and RIT and Cornell are doing. So we're really excited to be part of this. And through this project, we've got 1 year of funding now, and we're very, sounds very likely we'll get 4 more years and like $4 million more for this big program. So to me, that means like 1 grad student for 3 years, which I'm super excited about because that person will get to work on not only like measurement of potassium and nitrogen in fruit and measurement methods, but we'll also get to work on how does nitrogen really potentially affect wine stylistic goals and how we reach our goals in winemaking and sort of the concept of varietal character and terroir. So I'm super excited to have finally found a way to investigate this a little bit, but within the broader context of vineyard management, which I think we can all agree is probably the most important thing underlying wine quality. So I want to just thank all of the research sponsors. It's been a real jigsaw puzzle putting this together. The Virginia Wine Board has been a huge support, as well as USDA and Lallemand Scott Labs. So lots of different pieces to the puzzle. And of course, the people who do the work, the grad students and postdocs. And I want to thank you all for your attention and for the opportunity to present and to be here with you guys today.
Amanda, this is Michael Drash, winemaker at Stone Tower Winery. Great presentation, by the way. I had a question. Did you have a control with all those yans? Was it like at 21 bricks or 20 bricks or 24 bricks?
With the survey work?
Yeah.
No, it was just what it was. I mean, we just gathered data at what should have been commercial maturity, and I'll say should have been because I know in some of those research plots, there sometimes the grapes get harvested a little earlier than maybe commercial maturity, but no, it's just kind of what it was. And the vineyard manager, it was not in a one controlled research vineyard. It was
everybody measure.
Yeah, I just reached out to a lot of colleagues and said, hey, can I get a juice sample and measure the yan? Or in some cases, like the Cornell people had already measured it and they just shared the data and I pulled it, so I just wanted to get a picture because there was no yan survey from that part of the country.
And one other quick follow-up. You probably didn't do it, but it would be an interesting topic is the formation of methyl carbamate with too much, DAP and superfood type stuff,
Sure. And that's been, obviously that's been a big issue. I think UC Davis Extension has a publication on that with some recommendations, but I think in general, they're looking at like recommending not to add over like 400 milligrams per liter and things like that. But in our case, I don't think we're pushing that high usually, but in the Midwest, they might be. So it's a valid concern. But they also don't export to Europe. Not that we don't have the same health concerns here, but regulatory wise, I guess it didn't come down on us yet. So I think the momentum on that area of research kind of slowed down in the US because our regulatory didn't go there.
Thank you so much, Amanda, for presenting that information. And I would just thank you, Michael, for asking the first question for us. If those of you out there have a question, feel free to do as Michael did and unmute yourself and ask. If not, if you're in a place where that doesn't work so well, or if you want to just write into the chat, we'll make sure to look at the chat as well. So Amanda, I'm going to kind of start off with another question as we're kind of waiting for others to get their questions in there. You know, when we're looking, I will confess that most of what I know about nutrients, I learned from a catalog. And so I will say that it's really helpful to hear some of the real research background to that. But most of the time, you know, we're given guidelines about when we should be adding nutrients. So, you know, you should be adding this nutrient at, you know, 2 to 3 Brix depletion and this at 1/3 Brix depletion. Can you talk a little bit about the importance of the timing of nutrient additions and why we're given those particular benchmarks and what happens if we kind of miss them?
Yeah, sure. So to do that, let me put my slideshow back up. Okay, can you see my slides again now?
We can see your last slide, yes.
Okay, so I've got, after that, I have a couple of slides about that point. So let me move to, okay. So yeah, so the timing of additions really matters. So to kind of set this up, one of the most important things that a yeast cell is doing while it's trying to survive during fermentation is regulating the pH inside the cell. And in order to do that, it's going to be pumping out hydrogen ions. So this takes work, and then bringing amino acids into the cell also takes work with these proton pumps. And the cell membrane is allowing the cell to kind of balance those hydrogen ions, which are what drives pH. So as the alcohol level rises in the wine, it's not must anymore, I guess it's becoming wine, the alcohol level rises, the cell membrane loses integrity and becomes leaky to these really small hydrogen ions. And as they kind of seep into the cell, the intercellular pH will start to go down and get more acidic, and the cell is weakened by that. So it will stop using energy to pump amino acids in at some point around between 1/2 and 2/3 Brix depletion. I think most people would agree. When the alcohol is getting to I guess it's going to depend a lot on what your starting Brix was, but over like that 5, 7, 10 like percent, somewhere between 5 and 10, it's going to stop being able to regulate the cell integrity, and different yeast strains are more resistant to that than others. But in general, that ability to pump amino acids into the cell is lost because the cell is just trying to maintain the membrane and survive.
So if I am a winemaker who has the strategy that, you know, I'd rather not add anything unless it starts to smell bad, if that's happening, you know, after I've gotten to about 5 or, you know, well, if I've gotten past 1/2 Brix depletion, adding nutrient at that point probably is not going to do as well for me as I might think.
I don't think you're going to get amino acids going into the yeast cell at that point. They're going to be in the juice or must, and you know, maybe other bacteria or yeast might be able to use them, but probably not your wine yeast.
Can I add DAP at that point and have it be taken up?
It's more passive, but I don't think that it's going to help at that point either.
Okay. All right. Other questions out there for Amanda? I have lots of nutrient questions, but I want to give you all a chance. Coming in from Lee, he says, so just tons of oxygen at that point. So again, if you get to that point where you've got your stinky fermentation and it's too late to add nitrogen, just would oxygenating at that point help? And if you want to speak to that,
I think.
I don't want to put you in the corner of giving one.
Yeah, no, it's all right. I think you guys probably know more about the effects of that than I do, but just from my limited hands-on winemaking perspective, I think maybe you're just kind of aerating and blowing off some of those smells, which might do it, but it might also leave some other stuff in there that you'd rather prevent, some of the sulfur containing aroma compounds that you might not smell yet, but have the potential to cause problems down the road in reductive conditions. So yeah, maybe the oxygen will blow out the H2S, but you might end up, still with some things there that you don't want that might cause problems down the road. But I would also ask I see my friend Beth Chang on here too, maybe and lots of winemakers with much more experience than me that could answer that question better.
I do have one observation. In 2005, I did it. I was at Luna Vineyards in Napa. We did, in 2005 was a cool year, Bordeaux-like year for Napa, where the vines were not stressed out like 2004, but we did an experiment on DAP, basically Yans, right? So we did virtually little to no additions. This is all on same block of Merlot. We did virtually no DAP superfood. We did the recommended amount on the beginning Yan and based on bricks and all that stuff. And then we did double the amount. And obviously they fermented different ways. The highest level, of course, fermented faster, and we kept them separate for about 9 months. That the one that had the most also had the most bitterness on the finish. So that was one experiment, 1 year at one winery, but that was my experience.
I think that actually brings up another question that I hear from winemakers a lot, that, you know, there's a fair number of folks that are sort of pulling back from any additions that they feel like aren't necessary to do. And in some cases they'll say, you know, I'm not adding nutrients anymore because my fermentation pretty much finishes. I feel like I have fermentation security without it. Are they, even if that's sort of at a lower level than what the recommended amount of nitrogen would be, are you sort of missing out on some potential flavor components by not feeding enough nitrogen into the fermentation system?
My best guess would be, it depends on your stylistic goals. So maybe you could get more esters, in that case with certain aromatic amino acid additions, but maybe you don't want that, and I think that strategy is totally valid if your vineyard management and climate's pretty consistent, vineyard management's consistent, vineyard's established. It might just work great, but if you're getting fruit from a different place or things have been very different that year, it might be worth it to check on it, and see if things have changed a lot.
Other questions? I've got one from Jacqueline. Are there any changes in nitrogen content or nutrient content during the ferment if other populations of yeast or microbes die off that are not your fermenting yeast that can contribute to the fermenting yeast stability? So what's the effect of having other microbes in your fermentation, either potentially probably from the beginning of fermentation? How does that affect the nitrogen environment of what's going on?
I think that's a great question. I would hypothesize that they're going to consume nitrogen and maybe leave less for your yeast to use throughout the fermentation. If those populations get up higher than they would, if you used more sulfites or if you inoculated with a commercial strain right at the beginning, if there's more growing, those bacteria and yeast need nutrients too, right? So I think it would stand to reason that they're using it. I don't think that there's been a lot of research on this. And I think there's a lot of potential because nowadays we can do more microbial community analysis. We can do microbial ecology studies for like a fraction of the cost of what it would have cost to do those things 10 years ago. So I think that we're going to see a lot more answers to that coming in the next 10 years than what we know today.
Yeah, I've seen a number of papers looking at the microbial ecology of the grapes, but not really thinking about, or not really following that through to say, how is that affecting the fermentation nutrient at that point? But as you said, hopefully that will come. So great question, Jacqueline. Other questions? I have one other question that I've really been thinking about. When we're in the winery measuring DAP, if I'm measuring Yan in the winery, most of the time we're measuring using the enzymatic kits that go through the spectrophotometer. So we can measure ammonia and we can measure NOPA. And then we use a formula to determine what the Yan is. When we send those off like to a lab like Virginia Tech, we also get a number for arginine coming back. And I guess I have two questions. First of all, should we be thinking about or worrying about that arginine number? And secondly, when I'm looking at my Yan number, would it affect my nutrition strategy if, for example, I have a lot of ammonia versus NOPA, or I have a lot of NOPA versus ammonia? Should I be changing my nutrient addition strategy based on that proportion. I appreciate that you said that those really aren't correlated in a normal way, but if I have the numbers, should I change how I'm feeding my fermentation?
I think that's a great question. And I think that's something that's worth doing some experimentation in your own winery on, because I think that to make a really broad blanket recommendation on that point might not be, the best approach. It would be simpler if we could do that, but I think that's going to depend on vitamins. That's going to depend on, the yeast strain that you're using. I think that there's a lot more complexity going on there. In general, like we argue about targets for like total yan, and now you're asking about, okay, what should the target be for amino acids and for ammonium ions? And it's like, I don't know. I think in the cider world, we see that there's very little ammonium ion at all in apples. It's almost all amino acids that make up yan in apples. So my gut feel is I'm a little more comfortable with recommending a DAP add along with the amino nitrogen, because it's not there. And we know from the wine world, a little DAP isn't going to hurt probably. So, there's that, but with wine and with it being all over the place, like some grapes may have a lot of ammonium, some may have a lot more amino nitrogen. I think it's a very, very good question. And I wish that we had research that would really solidly recognize what are the other factors that interfere with this. Not that we want to recommend that every winery measure, B vitamins and resample, but at least maybe there'd be a way, kind of how we do like soil testing, or vineyard nutrient status, like an annual thing where you could just get a snapshot and say, okay, mine are low in B vitamins, so maybe I need, a thiamine nutrient instead of, just adding a whole bunch of DAP that doesn't have that in there. So I think some of the complex nutrients come in with those vitamins, and it may be the vitamins that are helping us and not the nitrogen itself. And that's really hard to parse out because we don't have that data. And, they're not things that are super practical for wineries to start measuring, but I think knowledge could help us maybe develop strategies that are practical. As far as the arginine measurement, I think if you really, really want to know an exact number of the yan, it's nice to measure the arginine, because the arginine molecule has 3 nitrogens on it and it will count as more. So many wine grapes, especially Vitis vinifera cultivars, are highest in arginine. That's the most prevalent amino acid of the 20 or so that are there. So it could make a difference of plus or minus 10 or 20 milligrams per liter at most. But, then I asked myself in my research, I've just done the pan NOPA and the ammonia numbers, because I asked myself, okay, if I have a more and more precise number, what am I going to do with it? If I don't have a target that's within 5 or 10 milligrams per liter to hit, maybe knowing that value just from the pan NOPA and ammonia is good enough for practical decision making and for comparison to other data sets that are out there. So I haven't gone to that degree of precision, I guess, in my measurements. But it basically, it's just giving you truly a more precise number on the true yeast assimilable nitrogen that's there. And it's not going to be much different than if you didn't measure the arginine. Does that make sense?
That does make sense. And I would agree with you that if it's only going to change it by 10 to 20, that doesn't really change a huge amount of our management. We have a couple.
And I got to say, that's a guess. I don't know exactly 10 to 20, but I think it's on that.
But it's not going to change it by 100, right? So
I don't think so. No.
We have a couple of other questions that have come in. We do want to kind of we need to be moving along quickly, but I don't want to cut off questions. So we're going to do these as quick as possible, but I realize it's complicated. So Kirsty's asking, we've heard that yan of 140 is sufficient for a healthy fermentation. That's a number that's been around for a long time. Is that still valid, or is there some research that's moved that number? Or is that still sort of kind of baseline plus all the other stuff?
I don't know. I want to show you something horrible. Do you guys have a second to look at something terrifying? Okay, so let me share my screen.
Are you going to make us afraid to make wine at this point? I mean,
no, you guys know what you're doing here. Come on. Are you guys seeing my screen now?
We just see your Zoom screen.
Oh, okay. Let me. Okay, how about now?
we see EC 1118 interactive effective met and yan.
So this is horrendous. This was from a project we did about 4 or 5 years ago on cider fermentation. And what we were looking at was, let me just set it up for a second. This project looked at trying to boost certain individual amino acids rather than just making a big dump of like a commercially available yan boosting product. And so my student dug into this and saw that like with grapes, there had been some research that methionine below like 20 milligrams per liter could lead to H2S production pretty reliably. But the thing is, Vitis vinifera wine grapes typically have way over 20 milligrams per liter methionine. They have generally around 50. So that really, that issue was identified 20 years ago. But people started to measure and said, oh, well, wine grapes don't have a problem here. Well, cider apples do. They're often below like 5 milligrams per liter methionine. So we thought, hey, maybe we add some methionine and we decrease the H2S production. So that was what this project was about. But the disturbing finding here, what this is like at different total yan concentrations, like this was what was in the apple was 53. And then we boosted the yan up to 153 and up to 253. Thinking that 140 is the target, right? So we did this, and then within each of those total yan levels, we messed with the methionine concentration. So we did like the control, and we added some methionine at 5, 20, and 50. So you can see in general, like for the control, adding a little bit of methionine decrease. This is H2S on this axis, so it decreased the H2S production. At 153, we had to add a little more methionine to get the effect. At 253, it really wasn't much H2S there, no matter how much methionine we had. But then you step back and you look, whoa, at 153, all the treatments had way more H2S than at 53. That shouldn't have happened. So
terrifying.
Yeah. So, and this was in a cider, just it was the White House juice. It's just a, juice, unpasteurized juice that was made in Virginia from a commercial producer. It seemed to ferment well in our controls and methods set up. So we used this, and I don't know what the B vitamin levels were in it. I don't know, I don't know. Maybe there was some other deficiency there. But then it really scared me to tell cider makers, yep, you need 150, because that was the worst, right? In this case. So I started to think a lot more about interactive effects, and we looked at some like post-harvest fungicides that are used in apple. We've looked at like storage, because apples are stored before they're processed a lot of the times. Grapes aren't. So there's a whole other set of factors to consider there in what could interfere with fermentation. But this really set me off, and I put this on here because I thought, if you guys asked me the target, I had to share this because I think this doesn't always happen, but it did. And we did this in all the replicates. He repeated the experiment a whole another set of replicates because, I saw this and I said something is wrong. Maybe you mislabeled them, but no.
Well, that's
so, and I think
terrifying note to end on.
But I think the 253, whatever was missing, that maybe boosted the yeast cell mass up enough to overcome, this stuff and do better. So I don't know. I mean, yeah. Sorry.
That's all right. Well, Phil, thank you so much. We do want to make sure that we have a chance to talk about the experiments that we have today. So we're going to thank you so much, Amanda, for sharing that with us. And hopefully we may, if we have some time at the end for some additional questions, then hopefully we can kind of come back to some of the other questions that are coming in. So with that, we are going to go and move into the wine flights that we had for today. So if you don't already have your wine set up, we'll start talking about the first experiment here. Let me work on getting my screen shared. Remember how to do this. Okay, so our first experimenter for the day is Rick Tagg. So I'm going to invite Rick to unmute himself. Rick, tell us a little bit about what was your experiment and what made you want to work on this question.
I wanted to see if there's a new nutrient called Stimula Chardonnay, and I wanted to see if it did anything. So and I wanted to try an experiment for the first time, so that's why I did it. Great. And what else do you want me to say? That's all I can say.
Let's tell the people.
The goal was to see if the goal was to see if it helped improve the wine, or actually if it did anything. And if it did do anything, what did it do? And it would be something we would want to continue using as a product.
Now, when you and I first talked about this, you talked about how you have 2 different Chardonnay products.
Correct.
So why did you choose the Stimula? What were you hoping it was going to do for your Chardonnay?
I was hoping, I was looking for we're looking for esters or thiols. I can never remember which one is which.
This one
but-
was esters.
Yeah, so we're looking for esters, and just to see what would, if it did with the advertised characteristics for the wine. But
Yeah.
Anyway,
So-
go ahead. Yeah.
So this is, I think, an example of kind of what Amanda was talking about, that some of the nutrients can have an effect on wine style. And so there's many of our suppliers now have specialized nutrients on the market. And as Rick mentioned, the most common ones are the ones that are supposed to be boosting esters or supposed to be boosting thiols. And we did have an experiment with thiols with Stimula Sauv Blanc, which is a thiol
Blanc,
booster a couple of years ago. This is Stimula Chardonnay, and it's distributed by Scott Labs. And basically it's something that the product literature will say that when you're adding this at about 1/3 Brix depletion, it's essentially adding in those precursors that then get converted into esters, and esters being some of our fruity and floral characteristics. So they're internal documents. The slide I'm showing you here just gives you the information really from the catalog, but when they looked at the difference between a Chardonnay, this is, I think, in a Spanish Chardonnay that was either fed with DAP or fed with Stimula Chardonnay, they saw an increase in some of these esters that contribute, for example, to rosy character, to they're showing strawberries and pineapples, so some of that tropical fruit character and some florals. And that was true both in the chemistry, they saw that in the chemistry, and they also saw that in the sensory. So the idea here was to say, well, if we use this on Rick's Chardonnay, do we see a shift there? But we did set up our experiment a little bit different than that. Rick, you want to tell them how you made your wine?
Okay, I can do that without getting too confused. So these grapes were from Nate Walsh's vineyard at Northgate, and I think they were picked. Well, I do know when I wrote it down. They were picked on the 17th of September, and we processed them on the 18th. I have 2 different numbers for the initial chemistry for the Brix. From Virginia Tech, I got 19.3, and from my hydrometer, I got 20.5. And you have all these numbers too, correct, Joy?
Yes, and I just realized I forgot to put the report into the chat. As soon as I get away from slides, I'll put the report into the chat. So if anybody's interested in like the nitty gritty of the ad rates and that sort of thing, that will be in that. For right now, we probably just want to think about how the general flow of things. So.
Okay, all right. So I'll keep going then. Anyway, so the fruit was chilled overnight. All whole cluster pressed, about 3/4 of a ton for each press load. And even though the fruit was really, really clean, my ultimate goal in this was to get a very aromatic, lean, kind of elegant style of Chardonnay. So more mineral and floral driven than fruit driven, kind of, if that makes sense to people. So I added a little bit of fermentation tannins, which was this is what I get when I write it all down, because then I can't even read it. With FT Blanc at the press, and it was I think 30 grams per hectoliter, something like that.
Yeah, it'll be in the report, so you don't have to worry.
Yeah, so I got it. I pressed it, kept it in the tank. Added the enzyme was added in the tank, and while the process was going on, there was I have one of those dry ice kind of producing CO2 guns. So I kept everything very, very blanketed during the pressing, and I pressed it. I mean, for how hard it was pressed to, I think maybe 21 PSI, could have been 19. So it was pressed pretty gently. That settled overnight. And because I was also using another product from the same company, I was to also avoid having to use so much sulfur, I put some Glutastar in the tank to mix it in real well as if we were pumping in for the juice. And then that settled overnight. So I previously was very well organized for once, and I got 2 barrels that were completely identical, completely from 2009, so completely neutral and clean. And actually it was 6 barrels, but for all intents and purposes, they were all the same barrel, same Cooper and everything. And so we used one of the things that, because we were also looking kind of anecdotally about the nutrients and the H2S production. So we very deliberately picked CY 3079 because that's a yeast, particularly for Chardonnay, that needs to be fed and pay attention to the nutrient requirements. So we barrel fermented that in the barrels, and somewhere along that time, because of the sulfur, the SO2, they're trying not to use quite so much SO2, but also trying to be being as clean as we could. I used some Bacillus in the barrels as they were fermenting, because there was no, Joy and I talked about, there's no possibility that we wanted these wines to go through ML, because that would change the parameters of the experiment. So that was an early first step to try to stop ML in the wine. So, and because of the Bacillus, I had to rack it. So I had to rack, and it was more than 2 lots, but that's another story. So I had to keep those lots separate and refill the 6 barrels. And of course, it was after fermentation, so the volumes were different. So I didn't end up with 6 barrels, and that was where I ended up with 2 barrels of the control and 1 barrel of the experiment.
But in the end, we do have one identical barrel, right? You're
correct.
Doing fine,
that
right?
is correct. Yeah, yeah. I'm doing all right. All right. And I have to say, this is the first time I've ever done this. I really enjoyed it. It's highly recommended. All right, I'm almost done, Joy.
Okay.
So they were sulfured. I think they were sulfured at 44 ppm, and they've been topped once since then. And so this is the ultimate wine. What it turned out.
Great. Well, and
Rick,
I appreciate you going through some of those details, because I think it does show that you did a lot of things on the front end to try to keep the aromatics of the wine and sort of protecting from oxygen. And then just to sort of go through the contours of the nutrient strategy itself. You did.
Oh, I forgot about the nutrients.
I'll walk you through it. It's fine.
That was the most important part of the experiment. So yes, I did an addition at 15 bricks, because Joy told me how much to use. I put Stimula Chard 40 grams per hectoliter in my experimental barrels, and Fermate O 30 grams per hectoliter in the control barrel. So, and we decided at the beginning that we were only going to do 1 nutritional add, because the Yan was 180, I think it was.
Yeah. So
actually, Yan was 210 to start. Yan was 210 to start. So
Rick took his Yan after the juice settling. So with white wines, you want to be taking it after your juice is settled, so that you don't get all the pulp in there. That was sent to Virginia Tech, and we were told that the Yanover was 210. So just to sort of get a real quick view of that nutrient addition decision, the Scott Labs catalog gives you sort of step-by-step. We know that we were at about 20 bricks, and as Rick said, CY3079 is a high nutrient strain. Scott will tell you sort of what you expect for your nutrients there. So the control in this case was just looking for fermentation security. And this table again comes straight out of the catalog. So for fermentation security, if we had 210 Yan to begin with, at our high nutrient needing yeast, our yeast, it says we would need about 250. So our add is about 40. So in this case, if we were sort of doing the low part of the add. So the low part of the add just recommended doing a 30 gram per hectoliter Fermate O addition at 1/3 bricks. But if our goal was to optimize the esters, which is why we were using the Stimula Chardonnay, then it recommended we use 40 grams per hectoliter at 1/3. So those are different rates, but that probably reflects some differences in the overall nitrogen contribution in those products. So in the end, we basically ended up, one set of barrels got a 30 gram per hectoliter Fermate O add, whereas the other set of barrels got the 40 gram per hectoliter Stimula add. So that's where the lay of the land is, so that you know what's in your glass in front of you. I will go ahead and read these off in case somebody has a really small computer screen or can't see what's going on. If you're in group 1, wine 735 was control, 273 was stimula, and 372 was stimula. If you're in group 2, wine 245 was control, 363 was control, and 940 was stimula. If you're in group 3, 941 was stimula, 107 was control, and 647 is stimula. If you're in group 4, wine 914 is control, wine 404 is stimula, and wine 954 is control. So we'll go quickly through the chemistry here. The chemistry was really fairly similar. So really those sensory results are going to be the most important. We can see from our juice analysis, we were given the NOPA, ammonia, and arginine. In this case, there was a high proportion of the arginine, but we see it doesn't really shift the YAN that much. So that part of that formula, I'm not sure how the formula works. I will look that up and send it out to you if you are looking at arginine, but most of us are just looking at NOPA and ammonia. So the fermentation curves themselves were fairly similar between the two. So that's one thing we want to look at with nutrient addition is, are we goosing that fermentation in any case? Here it looks like they went pretty much the same, and they both certainly finished with a residual sugar less than 1 with no trouble at all. The finished wine chemistry, again, was not much different. We wouldn't really expect it to be based just on the nutrient itself. But we did go ahead and measure the nutrients in the wine after fermentation. So this goes, I was really excited to see Amanda's data there so that we have something to compare that to. In this case, the overall YAN at the end is in the low 30s, and it's the same for both. So we don't see a difference there, and one of them leaving a lot more nutrient than the other. And in this case, it seems like all of the ammonia was taken up, leaving some residual NOPA and some residual arginine. I'm not sure exactly what that means. I guess I haven't looked at this very much, but I was just curious to see how much residual YAN we had along the way. So with that, I'm going to go ahead and come out of sharing my screen so that we can talk about here we go. So we can talk about our sensory a little bit. Let me go ahead and launch a poll here and ask if you were you able to tell the difference between the wines? So, and if you were able to tell the difference between the wines, I would love to hear if there were any descriptors that seem different to you between the wines. And so maybe you can either unmute yourself or you can write into the chat if there were some differences that you picked up between those wines. So we do have a fair number of people that have differences. So those of you that are answering yes, if you could give us a couple of comments there and just tell us what was different for you between those two wines. Okay. And if you haven't voted yet, I'll leave it open for just another minute or so. I see Melanie says that the stimula wine was brighter and fresher and a little bit more floral. So in that case, maybe part of what Rick was looking for in that. Chelsea thinks that the stimula Chardonnay wines have a fuller richness and more in the mid palate. Jacqueline feels like the control was less aromatic, so that goes along with sort of the flip side there. And then Kirsty said the aromatic intensity was higher in the control for her. And then we see Beth also felt that. So we have some differing perspective on what was different between the two wines. Okay, Shai had a much longer lingering lemon citrus on the aftertaste. Shai, with which one? And AJ, where the wine landed on the palate was different. So the stimula wine landed in the middle of the palate. And again, I think that there the winemaking goal was probably, Rick, you said, to sort of have a cleaner, crisper, leaner wine a little bit.
Right.
Megan, the stimulant one was more aromatic and had a rounder mid palate. The control was more minerality and leanness. So again, sort of the difference in style between those two. So let me go ahead and end the poll here and share the results. And I will say, I felt like these wines, Rick, when you and I tasted them before and today, I feel like there are subtle differences between them, but they seem to be fairly subtle. But you all were really good at picking them up. So
They
we
are
have about
really good.
67% of people saw the difference between the wines. So you know, I'll run the statistics on that, but that will turn out to be statistically significant. So Rick, how did you feel about the differences between the wine? Did you feel like they were different, and were they different in the direction that you wanted to go stylistically with this?
Well, that's an easy question to answer. When I tasted them right after they've been racked with salt, you know, I'd give them a little time to set. There was a difference, and I got more fruit, more tropical characteristics from the stimula, rounder, creamier. And then with the ferment O was more of a more minerality, more citrus fruit type thing, but so subtly different that in the long run, I wouldn't know which wine I would prefer if there was one that I would prefer. But today, knowing that and when you and I tasted them, and I've tasted them a couple times since then, the more I tasted them, the more similar they were. And I didn't think there was that much of a difference. But when I tasted, and I couldn't tell the difference. My original thought when I thought there was somebody who had left soap in the glass for the control because it was so much more aromatic. And then when I went to taste, I did get the difference, but then I went back with the note. You know what I'm saying? Is my decision on which wine to pick as the odd man out was driven by the aroma and not the taste. So,
Gotcha.
but I think that it's a good experiment because there is enough, a little bit of degree of difference that the idea of this experiment was to see how well the product works and also to see if it did make a difference. And I think that the difference that it did make when I do blend those wines together might make a better wine. Certainly a little bit more complexity to it than it had before. So I think it was worthwhile to do the experiment. Can I keep talking, Joy?
You can go ahead. I'll wrap us up in just a minute, but you can share your last thought there.
Anyway, so what I really enjoyed the most about doing it was actually paying attention to every step that I was taking instead of just because it's harvest and everything is usually kind of a reaction instead of a planned event. So that part of it I thought was really, really kind of interesting. So, and I think the wines turned out pretty well. So anyway, that's all.
Well, that's great. Well, and we're thankful that you did it. We have a couple more comments that have come in. So Megan says, when Rick sent me the samples last month, I couldn't tell the difference. And I think Megan and Rick and I have tasted this wine several times, and I do think I
need to say one more thing.
Every time.
As I tasted it before I got on, and there was a difference, but I couldn't tell what it was. The longer it went on, the more similar they were. So if that's my other grain of salt with it.
So Wendy says, as like last time, the two together is best. And I think that goes along with what you're saying with blending. Kirsty is curious to see what would have happened with no nutrient addition at all. And Kirsty,
I think I might be trying that next year.
I think that's an excellent question. And it would be a
really good question
to set up. So Kirsty, if you're interested in that, or if others are interested, we would love to sort of have that question a little bit to say how much nutrient is really needed.
And I'd like to see a difference between a solely DAP driven experiment and maybe Fermate O, Fermate K kind of, or any other company product. So
you know what happens when you do that, Rick, is you just signed up for some experiments. So thank you.
Well, I know I'm going to talk too much, but I really enjoyed. I mean, for me to take all these notes and actually focus, it's a miracle. So, I mean, and I think that ultimately, with the whole purpose of doing things like this, it makes us better winemakers. So, because we pay more attention. So, attention to detail as it's possible really helped a lot. It helped a lot. So,
well, thank you for doing this. We do have the result of our poll here. So, I asked, do you feel like the stimulus Chardonnay, or basically, do you feel the use of stimulus Chardonnay achieved the better winemaking or the winemaking goal? About 61% of folks did feel like it sort of moved it in that direction of your winemaking goal. And when we asked about preferences, it's split evenly. So I think that has to do with wine style, right? We all have different styles along the way. So so we want to make sure to have some time to talk about Michael's experiment. So again, Rick will be around at the end too if you have additional comments or suggestions for Rick. So at this point, let's go ahead and switch over to flight 2. And as you do that, I will go ahead and put the reports for both of the experiments in the chat right now so that I don't forget to do that. I'm sorry, I forgot before. I'm gonna go. There we go. Okay, and then we'll go back here. Oops. And then let me go ahead and share my screen again. So Michael, why don't you tell us what was your experiment and why did you decide that you wanted to work on this question?
Sure thing. First of all, I'd like to say thank you, Amanda, for your presentation. It was fantastic, but you're leaving us with more questions than answers. And Rick, that was a great trial. Thanks for sharing your Chardonnay with us. So just to talk a little bit, maybe before I talk about my experiment here, I just kind of wanted to talk about where I'm coming to this from. So I came of age in Virginia wine in the mid-90s when we were transitioning from really nobody measuring yan and having very cookie cutter approach of either adding nothing or adding the same thing to every single wine, with a lot of work that Bruce Acklin from Virginia Tech did, as well as Lisa Vanderwater from the Wine Lab in California. She came out quite a number of times. And we transitioned to where most everyone was measuring yan and making additions, targeted additions based on Brix levels, breaking it up sequentially when it was called for. It was through my work with method champenois production that I started to look at yan post-fermentation just to see if I would need to make an addition then. And I was a little bit surprised to see some of the yan levels. They were much higher than I would have thought. So that made me kind of back up my thinking to maybe question some of the additions we were making during fermentation, particularly with red wines where there's, we have a very short window between when the grapes arrive and between that, 1/3 Brix completion when we're recommended to make our final addition. So in order to be able to pull the sample, have the sample tested, decide what the ad would need to be, and executing the ad, there's 3, 4, 5 days there. It's not a lot of time. And when we're measuring the yan on red wine, oftentimes we're pulling the sample either that evening or in the morning. And my feeling is that our sample is perhaps not fully representative of the potential yan in there. And Amanda, I don't know if we're gonna have time to kind of address that question with you, but I was really curious about the timing of pulling the sample and the yan results that we get. But my feeling has been with red wines that there's maybe more potential yan to be used by the yeast than we're being shown from the lab. So so the sample we're tasting today, this is a Tannat from Heny Vineyard from 2020. So the treatment and the control, all the winemaking was the same, except for there was a wine that did not get DAP and a wine that got DAP according to the recommendations that we follow. So we work with the recommendations from the folks at Superfood. So the yan that we measured in our lab on the Tannat was 170. And for a 23 Brix fermentation, they recommend a total YAN of 250. So we would, if we were looking to make up that difference of 80 parts per million between 170 and 250, we would use the superfood, which is a DAP amino acid blend, up to 0.48 grams per liter, 48 grams per hectoliter. And so that would give us like 48 ppm. So that would get us most of the way there. And then the remainder of that was would have been made up within DAP. So so the experiment's pretty straightforward. The exact same two wines. One of them got a little bit of DAP and one of them didn't. In retrospect, this isn't, we're talking about a pretty moderate DAP addition. So, there's not really a great difference in the way the wines were made. So that's what we're tasting today.
Great. Well, thank you, Michael. So yeah, as Michael said, this was a pretty straightforward experiment, just 2 T-bins of Tannat. One of them got the full recommended addition. One of them got the superfood only with no DAP. One thing to realize about superfood is that it does contain a certain amount of DAP itself. And so this is really the recommendations from the manufacturer of superfood, which is BSG. Depending on how much of an addition you need to do, they'll kind of just give you how much superfood and how much DAP at each of the additions. So sometimes it's just superfood the first time and then superfood plus DAP in the second one. Sometimes you're adding DAP in both. And really that's to get it to the required, the recommended nitrogen addition at that time. So I did launch a poll during that time. So if you haven't had a chance to answer that, that's still up and we'll look at it in a minute. But I was curious also with Michael's question, Amanda, do you have any advice to give about when we should be taking our nutrient samples for our red wines, given that, we're processing into our bins? When is the best, most representative time to get that YAN number?
I think you're at this point, your guess is as good as mine. I haven't seen any studies on that. That's something I'd love to incorporate in this longer term project, sampling for both YAN and for potassium, because, with white wines, we're pressing. So like if we get a sample in the lab of fruit and we press it, I think it's more representative of what you're fermenting. But with red wines, that's dynamic. And when I was working as a part-time harvestingologist, we would do it like the day after crush. And is that what you guys do mostly? Like most people the next day or 2 days, or I think it's kind of an arbitrary best guess, but I think it probably is a dynamic, like an extraction, right? Because you're probably extracting some from the skins and it may be changing. I don't know the timing. I think that's a great question though.
Well, we would love if you do some research on that, we would love to see the results of that. I think, when we try to make a recommendation for when to do that, it's kind of as long as you possibly can wait until the yeast go in, just to see if you've gotten as much extraction as possible. So folks that do cold soak, you want to take your YAN after your cold soak before you put in your yeast, because that's what your yeast are actually going to see. But I think, Michael, you have a good point there to say, are we getting everything there? So
and given to, I think also it's something like you want to be consistent, right? So whatever you're doing, if you've been doing this for many years and you're sampling after day 1 or day 2, you're going to develop your best practice based on that. So I'd say, don't suddenly change your sampling regime. If you've got it dialed in and you're getting the results you want, maybe, there's something to be said for that, because like Kirsty's question brought up, and a lot of us have mentioned, those targets are kind of, still in development and they depend on a lot of other things. So if you've got, maybe your 120 is somebody else's, 130 because they soak for, they take the sample right away and you soak. So I just wouldn't mess with your sampling regime unless you had a really good reason to, if you've got things dialed in,
It's good advice. So Michael did ask that we take a poll on these questions. And so before we sort of jump into the results here, it does look like most of you are, a small majority are always measuring YAN, and then Most people are sometimes or occasionally measuring yan. So in terms of what kind of information we go into that. And then in terms of how people are handling that yan addition, it does seem like we're all over the board about that. So thinking about whether we add exactly what the catalog says, or what experience has taught us, or basically work with what the fermentation is giving us. It seems like we've got we've got a mixed bag in that sense. So, okay, so we'll get back to our experiment here then. So again, we have one bin that got its full addition and one bin that got its just didn't get the DAP addition, so that you know what you've got in front of you. If you are in group 1, wine 966 had DAP, wine 396 did not have DAP, and wine 459 did have the addition. Group 2, wine 913 was no DAP, wine 219 was DAP, wine 511 was no DAP. Group 3, 117 was DAP, 499 no DAP, 233 no DAP, and then group 4, wine 256 no DAP, 112 DAP, and 282 had DAP. So we'll go through the chemistry here. Again, the chemistry we wouldn't expect to be very different, and indeed it really wasn't. We saw that the fermentation curves looked very similar between the two. We do have a little bit of a different temperature between these two, but that may be more about where the T-bins happen to be on the crush pad. It didn't seem to affect our fermentation curve really much at all. Then when we look at the chemistry here, we do see most of the chemistry is pretty much the same. We do see a little bit of a difference in alcohol here, and I guess that's another question for Amanda, and I keep asking Amanda these questions right after she's tasted some wine, so I'm sorry for that. Amanda, would there be any logic in seeing a difference in alcohol conversion with nitrogen availability, or is that maybe, I mean, it's just as likely here, it's a T-bin addition of sugar, so there's, we're estimating volume and everything else too, but would there be anything there that we might see a difference in sugar conversion?
Well, I mean, if the fermentation went to completion, I mean, nitrogen additions can affect whether fermentation goes all the way to completion. So, but these are small differences you're seeing here.
And the residual sugar was below 1 for both of them. So it did go completely dry for both of them. So.
But if you measured, like if you really precisely measured the fructose, glucose remaining, you might see a little difference there.
Okay. And then again, we did our chemistry after our nitrogen chemistry afterwards, again, because one of the concerns there was how much extra nitrogen do we have around, for example, for microbes to metabolize during aging. This is a tannat, so it's going to kind of be in the barrel probably for a fair amount of time. And in this case, it seems like there's very little left over, especially if we take some of those numbers that Amanda shared earlier as kind of averages. It looks like there's very little left over. Again, the ammonia is completely gone, and most of what we have left are in the NOPA category, so amino acids at that point. So. But again, here the question is more sensory than it is chemical. So let me let's see. To remember how to get out of here. There we go. Stop the share, launch the poll. So let's see if we could, if you were able to tell the difference between the wines and if you had a preference between the two wines. So, and same thing here, if you were able to tell the difference between the wines, I'd be interested to hear what you felt were the differences there. And maybe Michael, we can come to you. How did you feel about the wines themselves? Did you feel like they were different? And if so, what differences did you pick up?
Yeah, so I was not able to tell the difference between the wines, or rather the difference I thought I perceived was wrong. So yeah, so I guess I wasn't able to see a difference. So I think kind of what's really interesting to me is looking at the yan numbers after fermentation, and to me we're very low on both of those. So it makes me less concerned that there was an unnecessary addition there. I'm not seeing any these kind of negative effects, and looking at some of the chemistry, as Joy and I have talked about, is a little counterintuitive to me, in that I would think of a DAP as addition as pushing the pH up some, but we're actually seeing the inverse there. So that comes off a little bit strange to me, but I guess doing this trial kind of gives me maybe some little confidence, more confidence that we're not over adding in this situation.
Yeah, I mean, I feel like the amount of DAP that you're adding probably isn't enough to shift the pH too much. So I'm not sure what's causing that shift there, but I hate to bring this up because I know that your lab techs are on the call, but you do have the ability to look at, I mean, you could kind of pick out a couple of lots at the end of fermentation, each year, and just run NOPAs and ammonias on those afterwards, if that's part of, if you have leftover kit, for example, at the end of the year, or at the end of harvest, that might be something to do, just to sort of give yourself a little bit of peace of mind that you're not over adding. So, and I will apologize to Jess and Audrey, who I know are listening, and I just volunteered you for work, so I apologize for that. But you can tell me how you feel about it later. So, okay, so any other, let's see if we've got any, here we go. Now I'm starting to get some comments on differences here. So Melanie felt like the DAP, the wine that had DAP had black fruit, was more astringent tannins with more complexity, whereas the one that didn't have the DAP was softer with a lighter mouthfeel and less astringent tannin. So like a little bit of a shift into the richer profile there. Other comments on differences, if you were able to pick them up between the wines. Let me go ahead and end the poll and share as well. It does look like we had kind of we did not have quite as much of a difference in this particular experiment than we did in the stimula one. So about 55% of folks were able to pick that out in a triangle. I will again have to do statistics on that. That'll be kind of right in the middle. So I'm not sure if that will be statistically different or not once I do the work. So, and in terms of preference, there wasn't a clear preference between the two wines. And again, in this case, we were thinking too about microbial stability over time. So, so Michael, if you'd like to, we can pull some microbiology right before you blend it, if you haven't blended it already. So we could take a look and see how we're doing in terms of microbial stability there too. So, okay. Any other comments for Michael or for Rick or for Amanda as we kind of wrap things up today? Any other comments? I do feel like I would just echo what Michael said earlier, that, I think sometimes when we start to dig deep into nutrients, we end up with more questions than we started with. But at the same time, Amanda and I spoke about this earlier, that just, the idea is, how do we make wise decisions as winemakers? Sometimes there's not one cut and dry decision, but rather what's the constellation of complexity that we can think through to try to make the best decision that we can. We did have a question come through. Mike, do you only use DAP, or do you use other nutrients?
Yeah, so we use superfood is the first thing that we go to, and Joy was talking about how that's a mixture of I'd like to have the experts help me get this straight, but a mixture of amino acid, nitrogen, and inorganic nitrogen in the form of DAP. So yeah, so that's our first go-to. And with that, I can't recall what the limiting factor is there, but we go up to 48 grams per hectoliter. So that would make up 48 parts per million of difference if we were short. And after that, we would go to DAP.
Great. Any other questions? So Shai, thank you, Shai, for putting this back in. This was from before and we couldn't get to it. So Shai's asking, is there a relationship to worry about for high YANs and biogenic amines? And I think that question goes to Amanda.
Yeah, that's a really good question. There's a researcher in France named Jean Salmon, and he has done a lot on that question. And I think that, you know, he's done a lot of work on that. And I think his lab would say yes, that it can be, especially with amino acids like histamine, which we know can cause sort of, a flushing and reaction if you're getting really high levels of it. I think that the question is, what's unknown is what are the alarm levels for biogenic amines. There are small amounts of a lot of things in food and wine included that even though we can show it's present, it may not be at the level that's going to cause a physiological reaction. And there's so much individual variation in terms of what level of biogenic amine is needed to elicit a problem in different individuals, among humans. So let alone the variation among wines that you're going to have naturally present. So I think it's a good question. I think the ethyl carbamate is the main one that people have focused on with trying to limit, like the EU is limiting DAP additions and like nitrogen additions with the limit of ethyl carbamate in mind. I don't think there's any specific limits on or guidelines on prevention of biogenic amines, but there is Dr. Salmon, it's spelled like Sam, like Salmon. He's doing some work on that, and you could probably find his papers and see a lot more information on where he's going with it. But I don't think it's led to any regulations or guidelines yet.
Thank you. Okay. So it looks like our time is up for the day today. So let me, once again, we want to thank Rick and Michael and Amanda for sharing your knowledge and your experiments with us today. We also want to make sure to thank the Virginia Wine Board for continued funding of the research exchange. And again, I will thank Jolie Thompson, who filled all of those little bottles for you all and got them out to you safely. That has been a godsend for me to have her to do that. So, and thanks to each one of you for coming today, for filling out your sensory sheets and participating in discussion today. This really does make a big difference in terms of getting great feedback for our winemakers for their experiments. And if you're interested in any of the ideas that came up today in looking more closely into nutrient management, I would love to talk to you more about that, either as a formal experiment for next year or just ways to kind of talk through some ideas as we look at this particular question. So thanks so much for your time today. I hope that you all have a great rest of your week. And if you haven't already done so, look for that email with your invitation for the April sensory sessions. So thanks again, and we will see you next time.
Thank you. Bye.